Thank you very much, and thanks to the organizing committee for giving me a chance to present. Again, my name is Ellie. I'm a medical oncologist at Dana-Farber in the genitourinary cancer group, and I'm a computational biologist. I run a cancer genomics lab both at Dana-Farber and at the Broad. I'm really excited to be here in front of this audience to talk about some of our work and some of the sort of more general precision medicine things that are happening in our space. There shamelessly is my Twitter account if you'd like to follow along. I'm actually in a quite fierce competition with some colleagues to accrue some followers. Here are my disclosures. And I actually would like to point out one of my disclosures because it's relevant for sort of more properly introducing myself to this audience. I think most of you folks who I don't typically get to present to. You'll see at the end there is some equity stake in Microsoft. I received 5 shares of Microsoft from my bar mitzvah in 1993. There's me. Look at that beautiful head of hair I had. It was now long gone and gray, whatever's left. I'll let you do the math in terms of what that gift turned into. It was great. So I went to college at Stanford in the late 90s, early 2000s. I studied something called symbolic systems. It's a mix of computer science with philosophy and linguistics. There's one very tried and true career path for folks at Stanford in the late 90s, early 2000s who do symbolic systems. We go off to start tech companies, make billions of dollars, and sort of retire at the age of my current age, mid-30s, we'll say. And that was actually my career plan as well. That was what I was intending to do. But sort of a funny thing happened during college. Some of my friends were talking about trying to create something from nothing, a camp for kids whose parents have or had cancer. Seemed like a really cool idea. At the time, I actually thought, oh, this is something I could do to help build something from nothing and then use that experience in my future career as a tech entrepreneur like everyone else. That turned into something called Camp Kesem, a camp for kids whose parents have or had cancer. So we created the first one at Stanford in 2001. We had 37 kids. I had, prior to that point, in retrospect, had lived quite a sheltered life and had not appreciated what cancer can do to patients, but also to their families. And so with that sort of profound inspiration and sort of humanistic experience, that's actually what brought me into medicine and specifically into cancer. Proudly able to create the initial camp at Stanford and then off to UCLA as a medical student, create a second Camp Kesem all the way through my training. I was on the advisory board for the one out at MIT up until recently. And fast forward a few years later, sort of what did that create for someone like me in terms of being in medicine? So created a bit of an odd situation because I found myself as a clinician. I see patients every Monday morning at Dana-Farber in the GU group up on the 11th floor of the Yaki building. I was a computer nerd and still am a computer nerd and sort of proudly embraced that. And realized over the course of my training how little of that was actually seemingly directly docking into my realities as a clinical practicing physician. And I was able to increasingly immerse all of this in the sort of what was happening in both the basic translational and really ultimately clinical sciences in cancer. And so given all of that, what I decided to do was basically create a field for myself. I called it clinical computational oncology and kept saying it over and over and over again until I could convince somebody to give me a job. So what is clinical computational oncology? In sort of, from my perspective, the idea is simple. The patient is my model system, and what I'm doing is applying technologies to these patients and their samples to make inference and discoveries. I like to write code to ask and answer questions about the data I get from these patients and make inference in that regard and integrate it with clinical outcomes and everything in between, and link that to the ever-growing space of molecular biology as applied to cancer, of which there are sort of massive amounts of exciting things happening all over everything that this room and others are doing. And so with that together, clinical computational oncology is sort of the integration of all of those things, an interdisciplinary science for medicine, in the same way that symbolic systems was an interdisciplinary science for computer science. And luckily for me, that this idea or this notion wasn't actually happening in a vacuum when I was entering this space as a postdoctoral fellow in, say, 2011 or so, because around the same time, sort of the growing sort of snowball that is precision cancer medicine was really taking steam. And again, I will very simply define it sort of too simply for this audience, but just to sort of put something out there, that precision cancer medicine, as simply put, is that if you have an advanced cancer, we apply a test to the tumors, looking for tumor-specific targets that we can drug. That list keeps growing and getting longer and longer and longer and more complicated, as we'll talk about in a little bit. And then we do a clinical action based off of this test. We link this to some drug or some test or some clinical situation we might not otherwise do. And that, simply put, is precision cancer medicine. And of course, this was sort of gaining steam around the time that I was thinking about doing something like this with my interest and skill set. And even more so, something else was happening. Now, I sort of, as is always the case, like to put up this slide because if anyone plays conference bingo, we all know that this is part of the game. But this sort of cost of doing a genome sequencing slide is relevant to me for a different purpose. Because I actually like to take this figure, I add one more axis to this plot, and I think about data points per patient and how much theoretical data do I, as a physician, need to keep in my brain at any point in time to properly care for a patient sitting in front of me in the clinic. And I can actually map my medical training onto this plot. So I started med school in 2003, and all the way through the end of my medicine residency in 2010, it turns out being a doctor is not that hard. All you really need to do is keep a few salient points of a patient's history and physical, their blood tests, and some imaging data points in your brain, match that to some very basic other pieces of data, and you can actually do a serviceable job of being a doctor. I shouldn't say that too loudly, I guess. I hope this isn't being recorded. But then something funny happened. I moved out from California to Boston in 2010, 2011, and did my clinical year both at Mass General and at Dana-Farber. And in both institutions, they were rolling out these, in retrospect, simple, but at the time very complicated tests, whereby we were doing hotspot genotyping of approximately 40 to about 100 mutations in about, say 20 to 40 genes. And these data were being delivered back to the clinician at the point of care. So I, as the first-year fellow, would get basically a very long text document dumped into the EHR with a list of genes, mutations, and yes or no. And with zero training, was expected to interpret these data, and realizing quickly that not just me, but the attendings I was working with also had zero training in all the stuff we were getting, but this was just sort of arriving at our doorstep around that time. And of course, now here we are in the middle of 2019, and things have gotten even more interesting, because now it is very feasible and readily doable to generate genomes, transcriptomes, exomes from patients at one time point, at multiple time points, and take all of this massive amounts of data to inform patient care and directly relate to discoveries that can help people. And so all of this together has created the momentum for a field of clinical computational oncology that can really accelerate these principles of precision cancer medicine. So with that as the backdrop, what I thought I'd do for the purposes of this talk is I'd say 30,000 foot view, discuss a few very specific questions that we're interested in asking both in my lab and in the community that I work with, and sort of hope that this helps inform some of the ideas and thoughts being discussed at this meeting and engage in collaboration going forward. So the first question we like to think about is a very sort of reductionist one. I have one patient at one point in time staring at me in the clinic, and I want to think about how can I use all the data in the universe from that patient and all the patients that have come before him or her to make the best treatment decision for my patient? And can computational oncology help to inform this point of care decision making using all the next generation sequencing that we have? And so I don't want to sort of revisit some of our earlier work, but I'll sort of very quickly sort of gloss over it. This was my first charge from Levi Garraway, my mentor and friend and colleague. When I started as a postdoc in 2011, he basically sort of in so many words said, hey, we're about to generate clinical exomes on a bunch of our cancer patients. Can you figure out some way we can turn that into a report so a doctor can understand what this means? Which seemed like a very simple project in 2011, but sort of clearly was sort of blissfully ignorant at the time. And sort of the first instance of a method to try to take that on was something we created called PHIAL, P-H-I-A-L, Precision Heuristics for Interpreting the Alteration Landscape. There's a very obscure Lord of the Rings joke associated with this that I will not go through here. But the idea was very simple. Given a list of variants, it would resort the variants based off of clinical and biological relevance using some very basic heuristics. The code was written by me as a postdoctoral fellow in my spare time, so I would say very confidently not industrial strength code. And we applied it to our first series of patients, and we thought this was really fun and neat. We also developed a database to go with it, a sort of actionability database to try to find genes associated with specific clinical actions. That was part of the heuristic that we developed for our project, which is through something we called CanSeq, which was an NSGRI-funded initiative for prospective clinical cancer exomes to use to inform patient care. And I'll say, of course, that pause at this point and recognize, especially in this room, and including the speakers that you've just heard from, that we were not alone. This is not a novel idea. There's many wonderful efforts happening in all sorts of places and spaces, some that were within 5 blocks of where we were, that were sort of unrelated to ours, some all halfway around the world and everything in between. I tried to capture some of the logos in this slide, but of course, this could not be exhaustive. This could not be representative of all of the wonderful efforts. And this is what we were doing in 2013. Our code was released back then, and we solved the problem, and it's been great, so thank you very much. I'm just kidding. So what happened? Well, we did all this, and it was sort of something a little bit disappointing sort of happened along the way. For all of, I'd say, in the midst of the hype cycle for precision cancer medicine, some of the first clinical studies trying to directly apply this to patient care came out that were a little bit, concerning. This was one study looking at sort of molecularly targeted agent versus treatment of physician's choice, and you can sort of see the very not different Kaplan-Meier curves in this context, not significant here. The Moscata study from a couple of years ago, which sort of had, in addition to some other sort of not so inspiring findings, demonstrated that only 7% of the successfully screened patients benefited from this approach. Certainly these were not, perfect studies per se. What is a perfect study? But as sort of initial first pass, how is this actually working in a controlled setting? This wasn't exactly the sort of game-changing kind of thing we were all hoping this was going to be. And I think another way to actually frame it is to sort of poke fun at ourselves, because that's what I like to do for myself personally. This won't be the last time we do this in this course of this talk, and sort of talk about what was the enthusiasm for precision cancer medicine at my home institution in the studies I was just describing, the CanSeq initiative, all that prospective exome sequencing. And sort of for all of the initial excitement and sort of rah-rah, we're going to do this and solve all of cancer, what actually happened? So we have, as part of the study, we had these molecular tumor boards where we had to go through and sift through all of the variants in the patients and have all the experts weigh in sort of a traditional molecular tumor board kind of fashion. You'll see there was this huge gap between our ability to sort of generate the data from an upstream perspective, like that file algorithm I described, to actually then translate that into something that would get delivered to the doctors. There was a lot of challenges associated with this, and a lot of, I'd say frustration from the people participating in the molecular tumor board to enable this kind of research. And so we made an intervention to actually shrink that turnaround time, dramatically. And so what we actually ended up doing was just stop having tumor boards. And people said, oh, thank you, now we don't have to come to these things anymore. We don't want to come to these things. And if there ever was sort of a sign for, this is not really working the way we hoped, I think this, to me, was the real tell. People didn't even want to come to the molecular tumor boards anymore. And I think that's actually not that surprising because, and perhaps in retrospect this is obvious, but at the time we didn't realize how hard the implementation of a decision support point of care molecular profiling study was going to be when applied en masse across large patient populations. And there are many challenges beyond the ones I'm going to list, but I'd like to focus on a few because those are, again, the ones that I think computational oncology can make a big contribution towards making some change. One is that technology in the clinic is evolving. I mentioned when we were starting when I was starting as a fellow, not that long ago, but I guess maybe it was longer than I think, we were just rolling out hotspot genotyping, and it seemed profoundly exciting or terrifying to receive a report with 40 variants on a piece of paper and a yes or no. And what we can do now is generate lots of large-scale omics data from patients that are not just DNA-based, but are also RNA-based. And of course, where we know this is going are data types and molecular profiling modalities that are well beyond, I'd say, garden variety somatic genetics. Bulk sequencing, single-cell sequencing, immune assays, multiplex IHC, other kinds of data that can all come from the patient. And this technology is going to keep happening, which makes even freezing these data types to actually perform a clinical study particularly challenging, but something we got to think about. Two is that knowledge keeps evolving. the knowledge that goes into these sort of curated databases of variants and what they mean and whether they're clinically actionable cannot stay static. And we perhaps thought they could have back in 2012 because things seemed more simple, but we actually can demonstrate how hard this is by simply revisiting our knowledge base that we had developed back in 2013. And when we did that a year or two ago and just said, how many of those things are still clinically actionable, things that we thought might be in some way, shape, or form in 2012, 2013, turned out almost half of them were no longer really relevant anymore. And not only that, there's probably a lot of other stuff that was relevant. And suddenly this knowledge base that we were sort of purporting as being useful was clearly not useful at all. And even more so, what I think was perhaps rapidly obvious to a few of us, but really hard to know until this was happening, was that the doctors on the receiving end of all of these tests needed a lot of help. And the reports we were generating for them were, for lack of a better way to put it, like genuinely incomprehensible to the average clinician who was expected in the course of their 15 minutes, while not wrangling EPIC or other kinds of nonsense we have to deal with, also interpret molecular tests full of information that we had no training on. And so these are challenges, but of course these are imminently solvable challenges. And that's what I think is so exciting about the future of computational oncology in this space. So can we actually help fix some of these things? So I think so. So I mentioned our interpretation algorithm efforts early on. The challenge at the time was taking whole exomes and finding individual variants that were actionable. This was a very I would say, first order actionability assessment. BRAF V600E equals RAF MEK inhibitor, sort of a very point mutation this equals action that. And what we know now is that the world's a lot more complicated than that. And we need to actually integrate not just the somatic variants, but also germline variants, not just the DNA, but also the RNA for either RNA-based expressions of mutations, fusions, or perhaps even gene expression profiles of immune scores and whatnot. And we need to go even beyond, I'd say, the concept of first-order genetics. So it's no longer sufficient to just find the BRAF V600E mutation. You need to find, perhaps, the BRCA2 mutation that also has an underlying homologous recombination deficiency mutational signature. Or if it's there, great, and if not, maybe that isn't actually relevant. Or you need to look for microsatellite instability in the genomics independent of the genetics. Or you need to look for overlap between the somatic variants that you're seeing and the germline variants to make sure there's actually a proper connection between those two things. This is a lot more complicated than just trying to find the BRAF V600E, which, again, in 2012 and 2013 certainly felt complicated. I know it was complicated for me, but now things are a lot harder. And when we actually tried to sort of revamp our entire approach and compare what we were doing in 2019 to 2013 applied to a couple of data sets, one in melanoma, one in prostate cancer, not surprisingly, we could find a whole lot more stuff in any given patient or across patients and actually sort of revisit the concept of actionability for, I'd say, the current modern era. The second thing is that we wanted to make sure we were adding the right kinds of content to the knowledge bases we were thinking about. And again, what we didn't want to do, because there are so many spectacular knowledge bases for clinically vetted actionable variants that exist, CIVIC, OncoKB, PMGK, I believe I'm blanking on the one from Cornell. There's many others. I had listed them in the prior slide, and we didn't want to reinvent that wheel. There's no point. They've already done that. What we were interested in was actually saying, well, is there a role for any preclinical data that we could actually properly insert into this space to inform some of the frontiers of actionability? And we think this is really important because something else was happening that was starting to concern me. I'll say, here's one example from something, and I'll try to be very nice about this. But this was from one study that had a knowledge base where they purportedly identified alterations in MITF associated with giving a RAF inhibitor in melanoma. And I thought to myself, this is strange. I looked at the reference and I said, oh, that's funny. That first author of this paper that they're referring to sounds really familiar. This is my paper, except they actually got the inference wrong. And the experiments that we had put into that paper were actually showing the complete opposite of what I think the actionability was being purported to be. And I think this is because it's really hard to curate preclinical data when trying to think about actionability. I did not need to look any further than sort of a misinterpretation of my own study to find that and sort of recognize that this is something that we need to solve. And so one thing, oops, where'd Nate go? Oh, that's his picture. Oh, well, Just imagine a very lovely gentleman sitting next to Brennan over there. That's weird. So what we've tried to do is actually, one, revisit the concept of actionability and spend more of our time, knowing again that there's so many other great things happening elsewhere, spend more of our time in the preclinical space and look for sort of more of the frontiers of actionability. And then in parallel to that, try to come up with more thoughtful ways of, in essence, trolling through PubMed and sort of using some more sophisticated NLP methodology, like some that's outside of the scope of this talk, but for which we just published in JAMA Oncology a couple of weeks ago, to actually try to find the right kinds of new actionability things in the literature in an automated way to help save our manual curators from having to do this. And when we did that, revisited our entire sort of internal knowledge base, we could inject all sorts of new content in a way that we probably couldn't have done previously, and also in a way that was much faster than it used to be. The third thing is okay, so if we think we can come up with sort of interpretation algorithms for sort of the modern era, we think we can inject knowledge that includes preclinical information in a more thoughtful way than we had done previously, is to think about, well, what happens when you actually deliver this to a doctor in real life? And one of the sort of most concerning things that I did over the course of the early sort of trying to think about this and really started channeling some of the spirit of who I thought I was going to be when I was in college as sort of a tech entrepreneur is I did sort of very old school usability testing, which practically means I printed out one of these reports. This is from our OncoPanel test. I see my friends at Brigham are smiling. Hopefully, well, it's all good, guys. Don't worry about it. And I basically printed out the report, and I started walking up and down the floors of the Yaki building at Dana-Farber, which is our clinical building, and I'd just pop into the clinician workrooms, and I would hand them the piece of paper, and I'd go to my friends, and I'd say, what does this mean to you? Very open-ended question. I got a lot of very interesting answers. So this is the printout of one of these example reports. What do all these colors mean? Those are copy number plot, in case you're wondering. Look at this actionable tier 4 mutation. So just so you know, the ontology of these reports, tier 4 is the only set of mutations that are not actionable. Which copy number events are important? Just so you know, in this patient, which you can tell by this sort of funky looking copy number plot, had almost every gene was either listed as being gained or lost in some way, shape, or form, and so the report went on for 20 pages. The last one I cannot attribute to my friends in pathology at my institution. It was not from one of our reports, and I will not shame the vendor who provided it, but I was shown a report that simply said EGFR. That's it. Wild type, mutant, amplified, deleted, nothing. No idea. It just said EGFR. And you know, that's not helpful. If you're on the receiving end of this, and you're a doctor, and you've got 60 patients that day, and you got a patient looking at you wondering, what does this mean with their New York Times article saying, you know, this so-and-so over here just got cured, why can't you do this for me? This is really tough. And so we actually tried to do something at Dana-Farber a couple of years ago. So again, almost like felt like a community service project, but I'm like, you know, I like to write code. This seems like a solvable UI problem, user interface problem in medicine. Let me take this report. We're going to basically create a web-based version of it that does not change a word of the content, but rather moves it around in a way to make it more intelligible, one, and two, adds sort of very simple kind of cues to help guide the person in interpreting it, and create a few fake patients and set up a survey study where we basically send this out to the doctors and we ask ask you some basic questions about doing this. And our only intervention is simply changing the nature of the report. And so what did we learn from this? Well, the first thing we learned is that not everyone really will respond to a $100 gift card. So in case you're wondering, the participants were surgeons, medical oncologists, and pediatric oncologists. Had almost 100% participation from pediatric oncologists, about 35% participation from med-oncs, 2 to 3% from surgeons. So in case you're wondering what that means, I will say nothing more on that. So what did we learn? Well, we learned a few things. Indeed, in certain circumstances, simply changing the nature of the way the report looked and helping the doctor figure out what this meant actually helped them understand some basic concepts, like tumor purity. If you have a very very impure tumor and you see no mutations, it does not mean there are no mutations there. It means you don't know if there are mutations there because the sample wasn't any good. And we could actually demonstrate that just by sort of creating a UI that actually highlighted this key point, very basic to perhaps this audience, but very confusing to the average medical oncologist, that actually helped the doctors make sense of this. That's the good side. The bad side is on some of the, I'd say, slightly more complicated things, treatment recommendations, for instance, the intervention didn't actually help. And I should add that these narratives, these fake patients we made, were not, I would say, advanced placement precision medicine. This was, your like melanoma, V600E, what drug do you give kind of questions. And what was really disturbing, frankly, was how many doctors got a lot of the questions wrong. And this is at a quaternary care medical center where we've been reputatively doing precision medicine since at least 2010. The other part that was kind of funny was actually at the end you get to put in some free text. You can say, well, just tell me anything else you want to say. And I have an Excel spreadsheet on my computer that has all of the sort of like all of the comments, and many of them have expletives in there that I cannot put on a slide. But what I'll just say is that in that document there was like sweating anger and frustration, because I think the doctors were so frustrated at this point from just basically for all of the reasons I just explained. And strangely enough, I would say the nice part was simply that the folks who were randomized to the web-based report actually were saying things like, oh, thank you, at least like, this makes a little bit more sense. So given all of this, what are we trying to do now? We sort of rebranded that file thing because Levi doesn't work at Dana-Farber anymore, and he's not my boss, so I can sort of take away that Lord of the Rings stuff. And we're trying to develop something that's sort of a work in progress. I'll say this is not, we haven't even finished the method really, and it's not even we haven't even put the preprint up yet, but one of these days we will. But this is the general idea, and sort of invite anyone and everyone who wants to help or has any thoughts to chime in. Whereby we take not just DNA, but information from DNA, but rather that from DNA and RNA, from tumor and germline. We inject all sorts of content. We have heuristics, but we also have sort of aggregate second or third sort of order omic features based off of the data we get. We have a whole other thing that I don't have time to talk about trying to match patients to preclinical model systems in sort of a matchmaker format. And we use everything we learned about basically the user testing on how do you deliver this content properly to create a web-based report. And basically create a one-stop shop interpretation method that anyone can approach and upload their data, run privately in their own accounts, that we never touch because that would be not good for anyone, and use that as a way to guide precision cancer medicine, as both a sort of web resource at moalmanac.org and actually a portal such that anyone, you basically set up a Google account and you yourself spin up a virtual machine, you run the method after you upload your own data and get your own results back one patient at a time. This is still a work in progress. We're still trying to work out the kinks on this, and especially the matchmaking part of it, which is beyond the scope of this talk. But again, I want to put this forward as, in essence, coding to help inform precision cancer medicine. This is sort of what computational oncology is, one patient at a time. We're also actually very keen to consider prospective trials for this because, again, you can't really deploy this stuff clinically properly until you actually do some real clinical studies. That is a whole other challenge that I'm not sure exactly how we're going to do, but we really want to and are eager to engage with anyone who has CLIA, whole exome, and transcriptome sequencing capabilities to actually make this happen. So I spent the first part of this talk really sort of focusing in on one patient at one point in time. And that is a lot of effort in clinical computational oncology. But I think we all know, especially for the metastatic patient population that we take care of, or the ones that I take care of, that even if we make the right choice and we do all the right things, it's probably not going to work for a long period of time. In some patients, that response or that progression to resistance takes a long time. In some patients, it's very brief. Every once in a while, you get these exceptional cases, and they just respond forever for reasons oftentimes we still don't understand. But this idea of looking serially at patients over time to understand clinical resistance is something we're very interested in as to whether computational oncology and sort of algorithms applied to these data in a longitudinal fashion can help inform resistance mechanisms that have immediate actionability for the next set of drugs you might want to develop all the way through to the next patient stratification schema you'd want to do. And my first foray into this, again, I think I actually hinted at this study in the prior section on the RAF inhibitors, was actually looking at melanoma patients, pre and post RAF inhibitors. We had paired biopsies from patients. We had robust clinical data, and I would say that remains the most epic problem. It's not actually the sequencing, it's the clinical data. That was a joke, but that's okay. The epic thing, okay. And use this to actually make discoveries. And I don't actually belabor the results of this particular study because it's like from 6-7 years ago at this point. But it was the first foray for me about thinking about patients in multiple dimensions, specifically a pre-treatment sample and a resistance sample with purity and ploidy-corrected mutation data, such that we could actually aggregate patients' mutations onto a simple plot and look for the cancer cell fraction, which is the purity and ploidy-corrected mutation data for these patients, and whether they fell into one of a few areas. The top right corner of this plot, which is basically clonal mutations that are shared in the pre- and post-treatment setting. The bottom right corner of this plot, which is mutations detected in the pre-treatment tumor but not detected in the resistant tumor. And perhaps most importantly, the top left corner of this plot, the genomic resistance candidate mutations, the mutations only detected in the post-resistance tumor, not in the pre-treatment tumor. And we started thinking about this with targeted therapy in 2013 with RAF resistance, but of course, I wanted to bring this up here to demonstrate how this paradigm applied to small numbers of patients can actually have a profound impact towards guiding therapeutic development across a number of different domains. So again, I'm a GU oncologist, so prostate cancer is what I spent a lot of time thinking about. And one of the next things we did was apply the exact same paradigm that I described just now, pre-treatment and resistance biopsies, whole exome sequencing, clinical annotation to understand the depth of response, to a set of men with advanced prostate cancer, whereby we had pre-treatment and resistant biopsies, and actually also in this case knew the site of biopsy, which is important for some of the confounders of these kinds of studies, like heterogeneity. And what was interesting, this is just an N of 1 example, whereby we saw in the resistant tumor focal amplification in a region of chromosome 7 that actually spans CDK6. This is in patients being treated with anti-androgens, sort of analogous to what we see in breast cancer, we're now starting to see in prostate cancer, whereby CDK4/6 appears to be activated in RB wild-type disease. And indeed, preclinical studies using prostate cancer cell lines confirmed that the overexpression of CDK4 or 6 followed by CDK4/6 inhibition actually did a wonderful job, which again, in an N of 3 patients in this context receiving enzalutamide directly led to combination clinical trials, enzalutamide plus CDK4/6 inhibitors in a very focused clinical setting. This was not 1,000 patients, this was very few patients, but because we had matched biopsies, it was actually not that hard to find the signal. So that is just a very quick vignette about an anti-androgen therapeutic, a targeted therapy in a different context, and using paired biopsies to drive clinical trials. What about pure biology? So this is from a study that we did a couple of years ago looking at chemotherapy resistance in germ cell tumors, a very rare phenotype. And what we found here was fascinating, we thought. So this is just an example. This is the copy number plot on the top of a representative germ cell tumor patient. Not that interesting, other than 12p gain, which is canonical for germ cell tumor. For all germ cell tumors, all have 12p. And we and everyone else were staring at plots like these and saying, well, I guess that's all we got. When we actually started to look at the allelic copy number of these patients, we saw something very odd in these tumors. There wasn't simply loss of heterozygosity. So I'll actually zoom in on chromosomes 3 and 4 to help walk you through this. Chromosome 4, if you look, actually has copy neutral loss of heterozygosity. You've lost a whole copy and it's duplicated itself. But chromosome 3 is something more odd. It has the same thing as chromosome 4, but then an additional gain, which basically adds another amplification event on top of a deletion event. And this reciprocal loss of heterozygosity that goes beyond LOH is something we had not seen before, and not nearly in the abundance that we saw as basically the defining molecular feature of germ cell tumors, as best as we can tell. There is no other tumor like it, sort of as the crux of this particular study. But I bring this up in this context because that same principle could be used to inform the tumor phylogenetics of this disease in the context of therapy, such that we were then able to observe over time and therapeutics that this loss of heterozygosity keeps accumulating. And this is not directly leading to immediate clinical actionability, but rather probably more to basic biology in terms of understanding what is the sort of, in essence, we think meiotic or meiotic-mitotic switch defect that's driving this disease fundamentally towards chemoresistance and differentiation, and what the generalizability of it is. But again, we actually made this discovery when it amounted to an N of 7 patients that we had serial biopsies in the context of chemotherapy, and some sophisticated algorithms to dissect this particular setting. Another context of chemotherapy, again, I bring this up in part because we spend most of our time thinking about targeted therapy and immunotherapy, which I'll come back to in a moment, but I just love to talk about chemotherapy because it works and nobody else seems to want to. We applied the exact same paradigm to bladder cancer, so I basically hit on, so far, prostate, germ cell tumors, and bladder cancers. I've gotten 3 out of the 4 cancers that I treat clinically. In this context, this was pre-treatment and post-exposure to chemotherapy prior to cystectomy. So this is a muscle-invasive, locally advanced disease where we had these paired biopsies. Initially here, we saw nothing. We saw no difference, nothing interesting at all comparing the genomics of the post-exposure to the pre-treatment tumors. And that's demonstrated by the figure on the bottom here. Then we actually started looking at some of the more, I'd say, second-order genomic properties of these tumors. So not just individual mutations, but in this case, mutational signatures. And here we saw something kind of curious. In the pre-treatment tumors, as expected in bladder cancer, we saw 2 dominant mutational signatures, ApoBec and nucleotide excision repair. In the post-exposed to platinum tumors, we saw those same 2 signatures plus this other signature kind of showing up. And we had no idea what this other signature was. It did not match any of the defined cosmic signatures at the time. But what it did actually match was a experimentally derived cisplatin exposure mutagenesis signature from some preclinical model systems. And in this fashion, we basically used the same paradigm of pre-treatment and post-exposure or post-resistance tumors to uncover a cisplatin-associated mutational process in tumors, only in the context of therapy. That, frankly, we don't still understand what the meaning of it is, but we now know, 2 or 3 years later, has been seen in multiple other domains, whether in solid or hematologic malignancies. Actually, it's evidenced by the recent Alexandrov preprint on in this space, which is really exciting for informing sort of questions about how do therapeutics mold the genetics of the cancers that we see in the metastatic setting. So all of this was leading up to what the only thing anyone ever really wants me to talk about, which is immunotherapy. And can we apply the same exact paradigm to an immunotherapy context and make some inference there? Pre-treatment and resistant tumors with sophisticated computational techniques to make discoveries. So this is an example of a patient with a uterine leiomyosarcoma. On the top, you can actually see pre-treatment pre-pembrolizumab. There's a big old mass in the liver along with one in the pelvis. There's the arrow pointing to it. On the right-hand side, you can see profound shrinkage of 1 sentinel mass. The pelvic lesion keeps growing. In fact, that was the only lesion that kept growing in this patient. And after 18 months on pembrolizumab, this was, I think, maybe the only leiomyosarcoma that's ever responded to immunotherapy, but happened to be this patient. This patient went to the OR. They resected that pelvic mass, along with actually the liver lesion, which turned out all that was left was sclerotic tissue. There was no cancer left. And we again had an opportunity of a pre-treatment sample in this case, intrinsically resistant tumor, to perform some of the same analysis methods I was just describing. And again, here, it gets even more interesting because we're not just interested in the genomic events that happen in the post-resistant space, what mutations are gained in that tumor, or detected in that tumor that were not detected in the pre-treatment tumor, but we're also kind of curious to wonder, gosh, are there any mutations that are not detected in the resistant tumor that are actually conceivably immunogenic? You just heard a wonderful talk about sort of the challenges and the cool stuff happening in neoantigen detection. Maybe that's actually relevant in this particular domain, in this particular context. And so as it happened in this particular patient, there was, surprisingly for me, only 1 non-synonymous mutation in the resistant tumor that was not detected in the pretreatment tumor, and it was in its second hit in P10. We could not declare from this N of 1 that therefore QED P10 loss leads to immunotherapy resistance. We're not making that claim, but it was rather striking in this case that this actually was sort of the only event that was in the resistant tumor. especially in light of the multiple preclinical evidence and sort of studies that have arisen since then that are also invoking PI3 kinase signaling as at least immunoresistance in some contexts, which is interesting. But again, we're also pretty interested in this bottom right-hand corner for this specific context. And indeed, this patient is alive and well and was able to give us all of her T cells whenever we wanted to. So we could actually figure out what the mutations were that were no longer detected. We actually had RNA sequencing on this patient as well. We could actually look for genes that were down-regulated as well on top of that, synthesize the peptides from this particular patient that were predicted to be immunogenic, take the patient's own T cells and do some pretty simple assays to go from a theoretical space of about a few hundred putative neoantigens down to only 2 that were actually immune-provoking to these patients' own T cells and also not detected in the resistant tumor as sort of a means of informing neoantigen prediction using pre- and post-biopsies that could complement all of the neoantigen prediction work happening, I'd say in single time point space. I thought that was pretty cool. So that sort of leads nicely into the last part of this talk. If I started this presentation thinking about one patient at one point in time, and I gave you a few vignettes just now about seeing patients over time, sort of how does longitudinal exposure to drugs, whatnot, inform resistance? What about thinking of ever-growing large cohorts of patients at single time points with lots of data and new ways of thinking about these data to further complement the inference that we're trying to do here and further inform this notion of precision cancer medicine? So for example, if you've got a whole bucket of patients and some respond and some do not, you don't have serial biopsies to do matched analysis, but rather you have single time points, can you use computational oncology techniques to inform response and resistance patterns using these data sets? And here, I'll sort of stick to the immunotherapy theme that I sort of left off with in the pre and post setting because it goes without saying, that seems to be sort of that is the dominant thing that's happening therapeutically in sort of the medical oncology space for this decade. And we were fortunate enough to actually make some contributions to how tumor genomics contribute to cancer immunotherapy response. We and others sort of made some observations I'll summarize here. So back in 2014 and 2015, 2 studies came out looking at CTLA-4 blockade and melanoma. And again, these are both sort of under this framework of single time point, bulk sequencing, clinical annotation of response or resistance. Both of these studies, ours was on the right, a group from MSK on the left, demonstrated this notion of increased mutations in response to therapy. The same group from New York also showed the PD-1 blockade response in non-small cell lung cancer. There was an association with mutational load. And a group at Hopkins showed the association of PD-1 blockade and MSI-high tumors, those, of course, being very high mutational load tumors. And the constant theme coming up here was tumor mutational burden is correlated with response to immune checkpoint blockade, regardless of the histology or even the, frankly, class of drug, CTLA-4 or PD-1. However, and I'll sort of attempt to bang my fist metaphorically here, we're sort of curious to see how that TMB concept is snowballed to where we're at in 2019. Because I will sort of, again, just pick on myself and look at our own data. This was our study from 2015, CTLA-4 blockade in melanoma. We had pre-treatment biopsies and clinical annotations of response. It is true that there is a statistically significant correlation in this context with higher mutational load and response to immunotherapy. However, there are many high mutational load non-responders and many low mutational load responders. I would say this is a correlation, but it is a weak correlation at best in this context. In fact, when we actually aggregated whole exome data on about 250 patients, I'll come back to the study in a bit, and just did some very basic sort of AUC analysis, this is a pretty poor biomarker for predicting response across a few common histologies. And I think it really articulates sort of the need of computational oncology techniques to think about these data sets and ask yourself the question of whether you're looking for biology and translational discovery, or are you looking for clinical biomarker development? Because there is certainly a lot of translational biology discovery to be done in this space, but if you just didn't even have any context for what that AUC was or what that graph was for, and just said, hey, is this a good biomarker? I don't think we'd necessarily be jumping up and down and saying this is gonna the one to rule them all on its own. And I think speaks to how hard of a problem this is. Because if we started in the, I'd say, middle part of this decade with tumor mutational burden, as a dominant molecular property associated with response to immune checkpoint blockade. And that's where I think a lot of the effort has really gone towards bridging that into the clinic. Here we are in 2019 with a lot of different features. And it's gotten to the point, and this was from a review we did back in March, which is almost certainly already out of date because these papers keep coming hard and fast, that you can probably invoke just about any single gene, complex, or signaling pathway as being associated with response and or resistance to immune checkpoint blockade if you sort of scour any recent set of publications in the last 3 or 4 years. And this is a really hard problem. And there is almost certainly lots of truth in all the studies in aggregate presented here, but there's also lots of challenges when we're thinking about these techniques applied either for translational discovery or for clinical biomarker development. And to really articulate this and sort of hit this home, I'm going to again pick on myself because I don't want to pick on anyone else and talk about something we've been very interested in and trying to figure out how to dissect what this particular example means, in this case, chromatin mutations and response or resistance to immune checkpoint blockade, specifically in kidney cancer, the 4th of 4 diseases that I'm responsible for treating in the clinic. So we actually, building off of our melanoma work, wanted to apply the same paradigm to kidney cancer. We're lucky enough to access a cohort of patients with metastatic clear cell renal cell carcinoma. These are patients who had already received front-end VEGF inhibitors, sort of bookmark that because I'm going to come back to that in a moment, who then went on to receive nivolumab, for which we had, again, robust clinical data. And I will just, again, emphasize that the clinical data aspect of these studies is far harder to pull off because of all the epic problems than really getting the sequencing done. To sort of emphasize that and sort of as a need in our field. We use this information, we stratified these patients in terms of clinical benefit, no clinical benefit. The first thing we wanted to ask was, well, does tumor mutational burden have anything to do with response in this context? And not surprisingly, because there really is no such thing as high mutational load kidney cancer, there was no association whatsoever with mutational burden and response to immune checkpoint blockade in this context. We did not see this. We did not see really any way we sliced it. We didn't see anything, clonal or subclonal, indels or mutations, really didn't see anything. Now granted, I should also mention, this was a tiny data set, relatively speaking. This was not like thousands of patients. And that being said, there was not even a wisp of a trend going on here to help inform that as sort of the most likely thing going on. So given that, we started to ask about specific genes and applying, in essence, some biological priors to these data sets. We already know a lot in the latter part of this decade about the genomics of kidney cancer. So rather than fly blind, let's apply some of the stuff we know and see if anything pans out. And when we actually focused in on the genes that we know to be relevant in this disease, there was something kind of curious. Even in this small data set, loss of function mutations in a gene called PBRM1 appeared to be associated with response to immune checkpoint blockade in this specific context. And nothing else seemed to pan out. This was a rather striking set of data in retrospective Kaplan-Meier curve. It was sort of not obvious, not subtle difference in terms of the outcomes stratified by this mutation class in this specific context. I just want to emphasize, we actually did a post hoc analysis of our aggregate data set from the study to look at whether there was actually whether there's any other confounders here. And I should just say again that in this context, the signal was being driven by the folks who had already been exposed to VEGF inhibitors, themselves immunogenic for reasons we don't totally understand. Those patients were the ones getting benefit with this genetic background, not the folks who were getting it upfront therapy. Just to really emphasize how confusing and complicated this is, that's what we saw. So what is this gene? PBRM1 is a member of the PBAF subcomplex of the switch-SNF chromatin regulatory complex. There are many genes. This cartoon, I believe, my friend Sigal Kadosh, her whole lab focuses on this complex, has actually added a third complex to this complex in the last 6 months, just to make this cartoon already out of date. But for the purposes of this presentation, this is sufficient. There it is. It's uniquely one of only 3 genes that are only members of the PBAF subcomplexes, because a lot of these genes sort of are promiscuous and actually form in various ways, shapes, or forms in multiple aspects of the BAF or PBAF. So this one is one of only 3 that is part of just the PBAF. The other 2 are BRD7 and ARID2. And I bring that up because here we are with this curious finding in the clinic, linking clinical outcomes to genomics. we could actually start to ask some interesting questions because we were interested in the translational potential of this observation. So collaborating with Bill Kalin, whose career and life's work is focused on kidney cancer and VHL, he actually had some isogenic cell lines, PBRM1 knockout and knocked back in. We actually found some interesting interferon and cytokine signaling in the PBRM1 null tumors, again providing some more smoke to this particular observation. In parallel to our work, and completely unbeknownst to myself, 3 floors below me in the Smith building of Dana-Farber, Kaya Werpefunning was doing an in vitro CRISPR screen looking for genes that, when knocked out, conferred T-cell mediated cytotoxicity in an in vitro co-culture model system, which was pretty cool. This is actually in melanoma, not in kidney cancer. These are actually the result of the screen. There's a lot, the colored dots that have labels are the ones that were actually genes that, when knocked out, conferred susceptibility to T-cell mediated cytotoxicity as a proxy for response to immunotherapy. I'll call your attention to the 3 red dots, because those 3 red dots were PBRM1, ARID2, and BRD7, which I just said were the only things that were just part of this PBAF subcomplex. Oh, that's pretty interesting. In parallel to that, unbeknownst to myself, David Barbie, another investigator at Dana-Farber and a friend, was looking at genes that, when knocked out, caused exposure of endogenous retroviruses, which themselves provoke an immune response, and indeed also found cell lines that had PBRM1 or ARID2 null settings tended to have more ERV exposure, all promoting this concept of there being real biology. Because if you go back to this cartoon, we were looking at PBRM1, we can actually now invoke BRD7 and ARID2. And maybe there's some real biology here. And I say that because I want to make a very clear distinction when doing these kinds of analysis between biology and clinical biomarkers. In our study, we were lucky to get access to a large-ish cohort of patients who had any kind of molecular profiling and response to immune checkpoint blockade in kidney cancer, could externally validate our findings. The vast majority of these patients, again, were VEGF-exposed tumors. That panned out. At ASCO earlier this year, independent of myself, other folks at Dana-Farber used our molecular profiling data set with clinical outcomes and again validated the same observation in a larger set of kidney cancers, a couple hundred of these patients. All is interesting. There's other work coming in terms of with phase 3 data that I can't talk about today. But I want to bring this up because our friends across the street, and some of whom co-authors with us in all these studies, asked the same question in a clinical trial for upfront immunotherapy, so patients who had not been exposed to any drug, who then got immune checkpoint blockade, for which we could stratify by PBRM1 status and saw no signal whatsoever. And I say that because this is a really hard problem. It's very confusing, and the biological observations we're using these clinical data sets to make don't always immediately translate into something that should end up in a molecular pathology report. Because this particular example is really confusing and it's really complicated, and it invokes a lot of biology and tumor-immune interactions that we do not yet understand. Also invoking a lot of therapeutic modalities that themselves shape things that we do not understand. And for which there is now a lot of understandable work and enthusiasm to figure this out, for which we'll need randomized data sets to validate and actually translate into biomarker status. But I will now, because I maybe wasn't as forceful back in 2015 with the TMB thing, I will put in bold and red an exclamation point and say, I do not think this is a clinical biomarker. And I bring that up because I is sort of curious to see, I've now seen some reports from commercial vendors who will, again, I find my own papers being cited in ways that I'm a little concerned about, perhaps over-enthusiasm in interpretation, when we're not sure the data really supports that for therapeutic decision-making outside of very specific contexts. Which again says that what we're really lacking here is sufficient data to make those inferences and make that leap from a biological observation that might have translational impact to a clinical, sort of immediately clinically actionable sort of event. And to try to tackle this, we sort of building on the momentum from our work in melanoma and our work in kidney cancer, we said, what if we just try to aggregate the world's molecular data of patients who received immune checkpoint blockade, single time point, stratified by response in various ways, and ask a sort of histology independent question. What shakes out when you actually look at this? And I'd say that sort of the first takeaway here was this was really hard. There's a big box of exclusions because these are, generally speaking, clinical samples. And clinical samples, as this room surely knows, come with clinical problems. Lots of FFPE blocks that have been sitting in somebody's desk for 10 years and suddenly need to be sequenced is not the same thing as TCGA. And indeed, we found a lot of instances, some of in our data, I'll be the first to admit, and some of the others, where there are lots of confounders and challenges using methods we did not have 5 years ago that could help sift out some of the stuff that could lead to issues, tumor and normal contamination, other kinds of sequencing artifacts and whatnot. So we removed a whole bunch of samples, created what we hoped was a clean data set, and asked a few basic questions. Don't have, obviously, time to go into all the detail on here, but I want to use this as an opportunity to demonstrate the potential of such an approach, because we're still at the potential stage, but we think this is really going to matter. So for example, here is the copy number focused analysis from this particular study of 250 patients. If you look at this plot, on the right hand, dots that lean to actually the right, or I'm not sure which orientation you guys. One direction is enriched for progressive disease, the other direction enriched for response. Most importantly, I want to very aggressively call out that nothing achieved even nominal statistical significance. 250 patients was not properly powered for the study. We were very, very overtly transparent about that, and we are woefully underpowered to really nail this question with this data set. However, we sort of saw a couple things that were interesting. Oh, look, there's CDK4 amplifications and resistance. That's interesting. We actually saw that in our data set internally. On the same day, all these papers came out in the preclinical space linking CDK4 and 6 to sort of dysregulation of the immune response, actually in a consistent direction, which we thought gave us encouragement that even though these data sets were underpowered, there's certainly knowledge to be gained that could inform biology and inform perhaps even target discovery, because that's, in essence, what we could see even though we were underpowered. So that's sort of a good example of how this could conceivably be used, but what about a more tricky example? We'll stick to the PI3 kinase stuff I mentioned earlier. There's P10 loss associating with resistance, even though, again, too few events in this data set to really make any proper inference, nothing statistically significant, but given a bunch of other stuff, like there's a study from our friends at MD Anderson, which had some nice preclinical data indicating P10 loss and resistance to immunotherapy and melanoma. Here's our work again, that I described earlier, invoking P10. Oh, this could be interesting. There's perhaps something to follow up on here. But I contrast that with the mutation data, which started to get really confusing. So there's a PIK3CA grid, and here are the mutations we saw in these patients. Now, the hotspot mutations in PIK3CA didn't associate with resistance to immunotherapy, but actually seemed to associate with response to immunotherapy. And on the surface, that doesn't make any sense if you think that P10 loss and PIK3CA activation should at least have some kind of commonality of activation of PI3 kinase signaling. How could one lead to resistance and one lead to response? Well, one explanation could be simply that these PIK3CA mutations aren't actually doing anything. And indeed, in this context, the PIK3CA mutations we were seeing were actually attributed to an APOBEC-mediated mutagenic process happening globally in these tumors, and just happened to result in these mutations. So all the clinical benefit patients, these PIK3CA mutations were arising from APOBEC-mediated mutagenesis and probably weren't actually functionally doing anything, because APOBEC-mediated mutagenesis itself is sort of a feature correlated with response to immunotherapy. And of course, the APOBEC mutagenesis tumors also had higher mutational burden, which itself correlates with response to immunotherapy, which is to say, all these features are confounded with each other at some level, and this is a really hard problem to solve, especially with 250 patients. And indeed, we sort of for future grant purposes, shot ourselves in the foot by including a power simulation in these data set and demonstrating we'd probably need a few thousand patients to make any proper statistical inference in this context, given the data that we just had in this data set. Which on the one hand said, oh, that's not good, Van Allen, you only got 250 patients, you're in trouble. The flip side is I bet within 50 miles of this building, there's probably been about 5,000 patients who've received immune checkpoint blockade in the last year alone, much less across the country, hundreds of thousands at this point. And this is actually conceivably a very solvable problem very quickly, were we all able to share in a more effective manner. And so this is a to-be-continued aspect of this space, but one for which we think there is major potential for everything from target discovery, biological development, all the way through to clinical biomarkers. which comes back to this concept of clinical computational oncology. Because if I started with one patient at one point in time and all the work we're trying to do to figure out how to make the best decision for that one patient, we follow these patients over time, hoping that we made the right choice, but knowing that more often than not, they're still going to become resistant. And we build ever-growing cohorts of patients with these clinical annotations to make increasingly sophisticated inference about response and resistance patterns, such that when the next patient comes in the door, we can do an even better job of making this prediction and do even better and really achieve this whole vision of precision cancer medicine. So how are we trying to inform this in, I'd say, the modern 2019 era on top of everything else I just described? One of the hats I wear at Dana-Farber is the computational director for our Center for Cancer Precision Medicine. And so an example, we're actually capturing standard of care immunotherapy-treated patients, now that there's a lot of them, and performing serial biopsy analysis, building up the cohort of single time point disease patients and also these serial biopsy cohort patients, doing bulk sequencing, but bulk sequencing is so, 2016. There's a lot of other cool stuff we can do to help learn and inform and augment the data that we know, including single cell analysis, preclinical model generation, cell-free DNA analysis over time. This is still a single institution approach to doing this, and this can never scale to the number of samples and patients one would need to actually properly do this. So to complement these kinds of efforts, we're now actually creating direct-to-patient initiatives that, rather than expect the patients to come to us for research, we bring the research to the patient's doorstep, quite literally. For example, I'm the PI of the Metastatic Prostate Cancer Project, mpcproject.org. Any patient anywhere in the United States and Canada could go to this website, click the Count Me In button, and we now have over 800 men so far in the first year, and we're growing. And so the way it works is you go to the website, you click a consent form. We then send you, that consent form gives us permission to send you a saliva kit, send you a liquid biopsy kit, access any otherwise discardable tumor samples, and access your clinical data, for which we actually had to buy a fax machine at the Broad to actually get the clinical data, which is a funny story for another time. Generate lots of data, and learn together as we grow. There's our liquid biopsy kit. The only place so far where a phlebotomist has given us trouble has actually been at Dana-Farber, which is pretty funny. I got a page about that a few times, and like, whatever, it's fine. If patients can't find one near them, we actually will send you a Quest voucher so the patients can get it done if they should not incur any costs for participating. What's key here is this is a way to expand research outside of sort of the quaternary care medical center system that we typically tend to capture. And indeed, 75% of our patients who have enrolled in this project are being treated in community settings outside of quaternary care medical centers who otherwise we would never be able to engage in research. Even more so, we mean it when we say this is not an academic hoarding exercise, and patients demand that it not be, and I think it's a silly thing anyways. And as fast as we can keep up with the data generation, we are depositing it in real time in a CBio portal, public CBio portal with full clinical data and molecular data. Here's a screenshot of our data release from January of detailed patient clinical information and the molecular sequencing. And even more so, it can actually help us answer some questions that I'm personally interested in a way that would otherwise be impossible. So, prostate cancer, other than Provenge, has no immune checkpoint blockade or immunotherapy sort of that's FDA approved, but we kind of all know, off the record, that these patients are getting off-label drug use, access to these drugs. And so the patients are self-reporting having received Pembro, Ipi, Nivo, and we now have these patients, and we can access their molecular data. We can send them biopsy kits, not just once, but as many times as we want, and keep getting serial assessments of their tumors while we're also getting clinical data. And in this way, ideally hope to build sort of an ever-growing real-world patient cohort across a number of diseases, prostate cancer and others, through our Count Me In initiative. So the last thing I'll say, I'm sorry going a minute or two over, is that the coolest part about, for me, is from a personal career satisfaction, is that I started this journey in 2010, 2011 with no track record for MDs pursuing this kind of line of work. Had to convince people, I had to make up a job field for myself, as I told you, had to convince people to give me a job, because this paradigm doesn't exist. What I think I'm finding, sort of with Dave and Diana were the first two brave people to join my lab, we now have a lot of MDs, a lot of whom have never written a line of code in their life before, who are making major contributions once they realize it's actually not that hard, and they bring to the table all the clinical and or biological domain expertise to really ask the big questions that matter. And in parallel, bringing computer scientists or quantitatively inclined individuals who otherwise would have no idea what the word resist criteria even means to actually make some contributions in a mission-driven way that matters. And I'd say from a personal satisfaction perspective, this idea that computational oncologists, clinical practitioners in medical oncology, radiation oncology, surgery, pathology, starting to actually do this, take this on in concert, hand in hand with the PhD computationalists together is what's really exciting for me on a personal level. So with that, I'll end. Thank my whole lab, the folks at the Broad I get to work with, the folks at Dana-Farber I get to work with, collaborators near and far, our funding sources, my wife for letting me escape these two little ones for about 18 hours. I'm happy to take any questions during the break, I guess. Sorry for running a few minutes over. Thanks so much.